hereditary angioedema or angioedema in general. And now we'll move to the second speaker, my dear daughter, Professor Dalia El-Ghoneimy. Dalia, she's a professor of pediatric allergy and immunology. She's one of the pillars of the pediatric allergy and immunology unit now. Dalia is a very active, fresh professor in pediatric allergy and immunology. She is a member in the Egyptian Society for Pediatric Allergy and Immunology, a member of the World Allergy Organization, a member of the European Society for Immune Deficiency. She's member of the Pediatric Rheumatology European Association, member of the Pediatric Rheumatology Internal Trial Organization, PRINTO. Dalia is going to speak about the pathogenesis of hereditary angioedema. You have 20 minutes, Dalia, for you. Free.
Thank you, Dr. Shireen, for your kind introduction for me. This is very generous of you. I will start sharing.
Questions and answers.
Bismillah ar-Rahman ar-Rahim. My talk today is about the genesis of hereditary angioedema. The objectives of my lecture is that one should, at the end of this lecture, one should be able to recognize the pathophysiology and genetic basis of hereditary angioedema with abnormal C1 esterase inhibitor, recognize the newly discovered pathogenic mechanism of hereditary angioedema with normal C1 inhibitor, identify the role of vascular endothelium in pathogenesis of hereditary angioedema, and this is very important to analyze the pathophysiology of hereditary angioedema, aiming for proper interpretation of the laboratory workup and understanding the mechanism of action of therapeutic options of this disorder. So concerning the pathogenesis of hereditary angioedema, whether due to abnormal C1 esterase inhibitor or those with normal C1 inhibitor, three systems are known to be abnormally activated during the attacks of this disorder. The context system, which is called plasma kallikrein kinase system, factor 12 dependent fibrinolytic system, the complement system, and the most important is the context system, together with the role of vascular endothelium in the pathophysiology of this disease. So let's start with what's going on when the context system is activated in normal subjects. Started with factor 12, which is got activated when in contact with negatively charged anions or proteases that is converted to active factor 12. The active factor 12 will then stimulate the conversion of pre-kallikrein into kallikrein. The kallikrein will then work on the high molecular weight kininogen, stimulating its the production of bradykinin. The bradykinin will then join its receptor, bradykinin 2 receptor on the endothelial cell and interact with the vascular endothelial growth factor and stimulate the release of nitric oxide and prostaglandin E2, ending in vasodilation, increasing vascular permeability and mediates pain. This does not end like this. There is a fibrinolytic system starting with plasminogen, which is then converted into plasmin under the effect of tissue plasminogen activator and to some extent under the effect of kallikrein. But the kallikrein stimulates the production of plasmin by stimulation of the conversion of pro-urokinase into urokinase that strongly stimulate the production of plasmin from plasminogen. And then the plasmin and the kallikrein induce positive feedback loop on factor 12 to produce activated factor 12. Activated factor 12 can be autoclaved into fragment factor 12. This step is first stimulated by kallikrein and plasmin. Factor 12 fragment is then acting on the complement system. It stimulates the autocleavage and activation of C1R and C1S, resulting into depletion of C4. This system, mainly the context system, fibrinolytic, and complement system, is regulated by the C1 inhibitor, which is serine protease inhibitor, that act as the main inhibitory step for context systems through inhibition of activated factor 12 and kallikrein, as well as inhibit the conversion or the production of fragment factor 12 and C1R and C1S, as well as certain other proteases of the complement system. Moreover, it inhibits the plasmin of the fibrinolytic system. There is another checkpoint for the context system is the degradation of the bradykinin by the angiotensin converting enzyme and the aminopeptidase 2. So what about the pathophysiology and genetics of hereditary angioedema with the abnormal C1 inhibitor? The level of C1 inhibitor are decreased, or the protein is dysfunction, and the net effect of C1 inhibitor deficiency. Therefore, the context system is inadequately controlled, leading to excessive release of bradykinin, which is the main and predominant mediator of enhanced vascular permeability in angioedema attacks. So this is a patient with C1 inhibitor deficiency hereditary angioedema. There is no checking point at the activation of factor 12, nor kallikrein, nor plasmin, or the complement system, or factor 12 fragment. Therefore, there will be marked increase in the production of activated factor 12, increased kallikrein, and therefore increased production of bradykinin, and the end results of excessive vasodilation, increased vascular permeability, and the resulting angioedema. There will be also increased plasmin and increased fragment factor 12 with excessive C4 depletion. And in this way, there is unchecked activity of the context system with production of bradykinin, the fibrinolytic system, as well as the complement system. But there is a very important point that this complement system does not share in the pathogenic or the pathophysiology cascade of the hereditary angioedema with normal C1 inhibitor. However, the depletion of C4 and other complement proteins like C1q represent a very important way for differentiation between different types of hereditary angioedema. So what about the role of vascular endothelium? The vascular endothelium can stimulate the conversion of pre-kallikrein high molecular weight kininogen to kallikrein, which is then act on the high molecular weight kininogen to produce bradykinin through the interaction with the brolyl carboxypeptidase and or heat shock protein. With this complex in the endothelial cell, stimulates the production of kallikrein. Once the kallikrein is produced, it will results in the positive feedback loop on production of active factor 12 and triggered activation of the context system. Heat shock protein, as we know, it's a protein expressed on the cells and in the eukaryocytes in general, and this can explain the attacks of hereditary angioedema in response to trauma and stress. Plasmin activation increases the fibrin degradation products, including D-dimer, that may be increased even in asymptomatic hereditary angioedema. So it is not strange to find the D-dimer elevated during the attacks of these patients. And although factor 12 can activate factor 11 and start the intrinsic coagulation cascade, however, hereditary angioedema is not associated with an increased thrombotic risk. There is associated as well activated fibrinolytic system, which puts things in balance. And as I have mentioned while demonstrating the figure, that the products of the complement system does not play a role in the development of the attacks. However, they are invaluable for distinction of C1 inhibitor hereditary angioedema from other forms of angioedema. So what about the genetics of this disorder? It is an autosomal dominant inherited disorder. The mutations are recognized in one gene, which is the gene that encodes 4 serine protease inhibitors, the C1 esterase inhibitor located on chromosome 11. Different mutations are recognized in this gene, resulting in 2 types of hereditary angioedema, type 1 and type 2. In type 1, the mutation results in truncated or misfolded protein that are incompletely secreted, leading to decreased plasma level of C1 inhibitor, which are less than 50% and generally commonly reach 5 to 30% of the normal plasma level. And while in type 2, this leads to fully secreted but dysfunctional C1 inhibitor protein. However, in about 20 to 25% of patients with abnormal C1 inhibitor hereditary angioedema, there is de novo mutation in this patient lacking family history, and this does not exclude the disease. So what about the pathophysiology? We are ending now the hereditary angioedema with abnormal C1 inhibitor. We are going now through the hereditary angioedema with normal C1 inhibitor. Several other genetic mechanisms were found to have a role in the pathophysiology of hereditary angioedema with normal C1 inhibitor. Recently, mutations were detected in the genes coding for factor 12, plasminogen, angiopuratine 1, myofiline, and kininogen 1. However, still in one or more remaining types of this disorder, the genetic cause is still unknown. So what about the hereditary angioedema associated with normal C1 inhibitor and where mutation was detected in the gene coding for factor 12? There was the promoter region of factor 12 gene shows similarity with the gene for estrogen responsive elements. Moreover, estrogen has been shown to increase the concentration of factor 12 in plasma, and that's why this disorder was thought initially to be X-linked dominant, but was found to have autosomal dominant inheritance, however, more predominant in females. Gain-of-function mutations was recognized that lead to overproduction of active factor 12. Moreover, the sensitivity of the mutant factor 12 protein is increased for the positive feedback loop and activation by plasmin, resulting in overly sensitive to activation for plasma and kallikrein and amplified pathogenic effect of the contact system. With the mutation in plasminogen, the plasminogen gene mutation may alter the protein structure and function. The protein may thus exhibit a higher affinity for tissue activators. This may lead to increased activations of the fibrinolytic system, subsequently leading to increased formation of the plasmin. The plasmin will exert stimulation for the contact system through activation of the production of activated factor 10 and the end results of increased bradykinin. This is a very important note that heterozygous, although it seems to have autosomal dominant inheritance, but however, heterozygous mutation in this gene alone was found to be not sufficient for causing hereditary angioedema phenotype, and these patients carrying heterozygous mutation were found to be asymptomatic. Therefore, this might need a second hit from another variant in the genes involved in coagulation or contact pathway. Another one is those having mutation in the angiopuratine protein. First, the function of the angiopuratine is to stabilize the blood vessels, promote endothelial cell survivals, and inhibit bradykinin and vascular endothelial growth factor mediated plasma leakage. These functions are achieved through the production of multimers. So therefore, angiopuratine should be assembled into multimers for proper interaction with its receptor, named tonica interna endothelial cell kinase 2, which is have the abbreviation of TI2 on the vascular endothelium. Therefore, this mutation results in normal total plasma concentration of angiopuratine 1 protein. However, the mutant angiopuratine 1 protein is unable to form multimers, resulting in vascular leakage. So the angiopuratine is not dealing with the contact system nor the fibrinolytic system. However, the mutation acts directly on the vascular endothelium, resulting in increased vascular leakage and accumulations of fluid in the deeper layer of the dermis of the skin. Myofiline is another protein that was found to prevent degradation of the vascular endothelial growth factor receptor 2. Therefore, they found some of these patients with hereditary angioedema, a normal C1 inhibitor, to have gain-of-function mutation, and found that they have increased localization of vascular endothelium growth factor receptor 2 to the plasma membrane, leading to increased signaling of vascular endothelium growth factor, therefore increased vascular permeability. The mode of inheritance was found to be autosomal dominant with incomplete penetrance with different presentation in different members of the family. The kininogen gene mutation was found as well in this group of patients and was found to result in production of an apparent bradykinin. This apparent bradykinin is functionally active, but may become resistant to cleavage by enzymes such as the angiotensin converting enzyme or aminopeptidase 2, leading to increased half-life and higher than usual activity. Then the mutation in the angiopuratine and the mutation myofiline and kininogen, they do not further activate the contact system, and the fibrinolytic system does not exert a positive feedback loop. However, they act directly on the vascular endothelium, resulting in the excessive vascular leakage and the pathological findings in patients with hereditary angioedema. In general, the role of vascular endothelium pathogenesis of hereditary angioedema, the vascular endothelium have an active role in regulating the plasma leakage in patients with hereditary angioedema. Several other proteins like bradykinin B2 receptor and angiopuratine receptor itself are not related to fibrinolysis and contact pathway, but directly involved with the vascular endothelium. And this is very important as it can open a door into knowing the different and other pathophysiological mechanism of hereditary angioedema, aiming for tailoring specific treatment for each disorder of this broad spectrum disease with broad spectrum underlying mechanism. So in summary, C1 inhibitor is the main inhibitor of several proteases of the contact system, active factor 12 and kallikrein, several complement proteases, and act as an efficient inhibitor of the fibrinolytic system plasmin. The contact system is the main system involved in the pathophysiology of hereditary angioedema, with production of bradykinin, which is the potent mediator of vasodilation and vascular leakage. Mutation in SERPING1 gene is responsible for hereditary angioedema type 1 and 2, resulting in decreased and or dysfunctional C1 inhibitor respectively. Recently identified mutation in the genes coding for factor 12, plasminogen, angiopuratine 1, kininogen 1, and myofiline are found to be responsible for patients with hereditary angioedema with normal C1 inhibitor. Vascular endothelium plays a substantial role in the pathogenesis of hereditary angioedema through several proteins and receptors regulating the vessel tone and vascular permeability, and this is a very good point of interest and further research to detect a specific mechanism of this disorder. And finally, I would like to acknowledge the pediatric allergy immunology team, our beloved unit that was founded by Professor Yahya El-Ghoneimy and now headed by Professor Shireen Reda. Thanks to all my professors and colleagues who are doing and always doing their best for the care of the patients. Thank you so much.
Thank you, Prof Dalia, for this very nice presentation. I think you tackled a very difficult and complex point, which is the pathogenesis with the different mechanisms, but in a very simple and straightforward way. Thank you very much. And now we move to the third speaker.